Hi, welcome back everyone online to the studio. We're just about to start the afternoon session. The music's quite loud behind us, but hopefully you can hear it and you can enjoy it as well. We've got three really special guests here today to start thinking about nanopore origins, which is one of the sessions we have coming up this afternoon. So I'd love to welcome to the sofa. We've got John Tyson, who's here from all the way from Vancouver in Canada. We've got Mark Akerson, who is here not only as a nanopore user, but also as a nanopore originator as well, all the way from University of California in Santa Cruz. And we've got Amanda Waugh, who's a Discovery Fellow at the Roslin Institute and the University of Edinburgh. Welcome, everyone.
Thank you.
We've invited you here today because you've been with us on the whole journey, from the beginning of MAP or nearby, all the way through to here we are today. And so I want to ask each of you, how is the work different that you were doing when this whole thing started to now today? Amanda, do you tell us about how the journey has looked from you?
Sure, yeah. So when I started, I was in Mick Watson's group. And he is a bioinformatician. He was developing bioinformatics tools for working with nanopore data. And there was no one in his group that was actually capable of producing data for him. So he was very keen for one of us to learn how to use nanopore sequencing so that he could run his tools on whatever he wanted. So he asked, would anyone be interested in going to PoreCamp, which John and a few others were running to help teach new people to use nanopore sequencing. So my first kind of experiences with it were learning how to use it on primarily metagenomic samples. And I think, PoreCamp was great. Everything went pretty well at PoreCamp. And then I got home and for 3 months, it just did not work. I spent 3 months trying to get it to work and it just would not work. So a major difference is that it does actually work now, and that it's staggeringly more accurate than it was then as well. So now I don't do as many microbiomes at the moment, but I've kind of diversified. There's a lot more kind of ultra long read sequencing, and methylated base calling, and all these things that we couldn't do near the start and with high throughput and high accuracy. So that's the biggest change, I think.
Fantastic. And Mark, for you, as someone who is really close to the technology, how has the experience of using nanopore sequencing changed in your views? And what's the differences in the kind of work that you and your colleagues were doing back then to today?
So the story about the origins is a different one, but beginning in 2014, our experience, like hers, was that we were trying to prove that the system basically worked. The throughput and the accuracy were on the border of being really useful. But now the throughput is through the roof. And the accuracy that we've tracked both in DNA sequencing space and RNA sequencing space is well beyond anything I ever anticipated. So DNA sequencing now, you can correct me, John, because you do this day to day, is that the accuracy is 99.5% on an individual strand. And we can sequence pieces of DNA, genomic DNA, that are 2 million bases long. People in Nottingham have done this. And so in RNA space and DNA space, it's now poised to do fundamentally important biology, like Carol Greider's talk today.
Yeah, fantastic. And I think we've only been able to iterate the technology because we've had input from the community, not just feedback, but also collaboration. And I know your team were working on bioinformatics and tools that could help us with data analysis. And obviously, John, you're working on wet lab methods. So what were you working on in sort of 2014, 2015 compared to now?
Yeah, I think the early days, what was like super interesting and fortuitous and great to be able to get on was, we were little groups spread around the world, and we were given the opportunity to basically, have a try of this thing, because it went out to a very broad community. It wasn't just in genome centers, big groups. It basically, the net was spread very wide, and basically, if you were keen and you really had an interest in developing it, you could do it. And it kind of, I guess, a number of people had the interest and the time to really get into it, and we enjoyed doing it. And it was like bringing together a community, and then from that kind of developed the kind of idea that actually we've been using it a little while, we've developed kind of our skills to do that. We should try and teach everybody to do that, and that's kind of where things like PoreCamp came out of. With like Nick Lohman, Matt Luce, Josh Quick, and Justin O'Grady at the time was obviously in an academic environment, and then Jared Simpson, people like that, where we thought we can get everyone together and we can start trying to help people just, I guess, not have to go through many of the pains maybe we did initially. Just kind of let them leapfrog that a little bit and try and get it to a more distributed kind of way of doing things. And so I think, the comparison, obviously in the early days, it was like, it was just trying to find those gaps. We were brought on effectively because we were doing, say, long reads sequencing of like transcripts. So we wanted all our splice variants put together in one molecule so we could see the context across the whole length rather than putting it together little pieces. But then that developed into many other things, like genomics and that sort of thing, just basically through the fact that we collaborated with people. And the people we met, we formed collaborations and then tried to push things forward and develop things as, obviously, and I think, it's never as quick as you'd like it to be, but generally you've got to imagine, or maybe it's just going to take twice as long as ONT say it's going to take, and then you'll be good. And it's like that kind of improvements that it's been astronomical in terms of it's getting what you see from the beginning to the end now has been very impressive, because when you think about that little device, basically it's this 512 single channel recording on a little USB stick. We have people like doing, they can't do certain things in some buildings where the grounding's not good enough to do it, but we're doing it on this little device that you plug into a USB. Technologically it's amazing.
I always think of the analogy of a mobile phone when I'm old enough to remember the first mobile phones, which, didn't have a very good signal. The battery life was about 2 hours and it was very expensive, but we still used it when we were out and about. But there was that one moment when you walked into your house and you thought, oh, I'm going to make my lasagna tonight. I'm going to make my phone call to my mum on my mobile phone. And it feels like that's where Nanopore is now, where it's caught up with those traditional metrics of speed, accuracy, and yield and cost effectiveness, having originally delivered long reads, native, real time, and now is providing more of the complete package for all of these applications. So you've described PoreCamp as sort of teaching the teacher model, and now the knowledge is spreading in a sort of more instructional way as we develop. We spend more time on our protocols and our instructional parts of our website and on our tools, but the community is still so thriving. So as you look ahead to the next 10 years, I know that you guys are now both interested in surveillance, pathogen surveillance. What does your fantasy future pathogen surveillance network look like? And would you ever collaborate between Scotland and Canada on that, for example?
Yeah, I mean, excuse me. Yeah, I think just early detection is really important. So there's a lot of groups now I've seen who are trying to kind of find environmental viruses that they can detect before anything big happens. And that would be really cool to kind of have a lot more environmental surveillance to get an idea of what is out there and better understand these things before they start causing problems in animals or livestock or humans. So yeah, I don't really know what that would look like. But it certainly helps to have a portable sequencer for that sort of thing, I think.
Yeah, and I think the opportunities now it's like finding those gaps or the unique capacities of, say, the platform. And for example, during say the pandemic, or we would tend to be if we really needed to push it in terms of very rapid detection of, say, Omicron arriving or Mpox, diagnostic kind of confirmation, that sort of thing, where we really needed to do it, we could drop to the Nanopore because we knew we could do a rapid library and then we could do the detection. By the time the muck scan's finished, we've got a detection, right, and we can tell. So it's like finding those windows of opportunity where it makes the real difference. And I think going forward, probably the lower throughput kind of diagnostic kind of platforms at the front line potentially, that initial detection, because it's all about the speed, I think. Where you've got to be able, you need to make a decision in a day or 2 days. Ideally hours. Then you can get something that's really quite actionable for individuals, and especially in the clinical. I mean, in the kind of public health and kind of environment or the clinical space. I think it's all about timing and how quickly you can do it, and necessarily maybe on a smaller scale as well, so it can be more distributed. I think rather than being centralised in some senses, depending on what you're doing.
I'm sure you're familiar with the work that's happening at Guy's and St Thomas's Hospital that's now being scaled up to 10 hospitals in the UK, where they've integrated real-time respiratory metagenomics into ICU. And in the trial, 3% of the results were notifiable to public health, as well as 50% of the results resulting in a change in antibiotic because of drug resistance. So it's absolutely the sort of line of first identification of those people who are getting really sick and understanding what it is and whether it's something new and unexpected. It's really exciting.
Yeah.
The idea about distributed sequencing for surveillance is great. So 10 years into the future, Mark, I mean, we talk about RNA a lot. If DNA is a teenager, is RNA a baby? Is it an infant? Is it a toddler? How much more is there to go for RNA science?
RNA is a rebellious teenager. And
Right.
so I think if you track this sequence identity over time of DNA, you can see a very sharp increase in accuracy. And the RNA sequencing accuracy is basically following that in time. And so it's been a sea change since the RNA sequencing was first introduced at 87% identity. And there's rumors that today there's going to be a new release on accuracy that's through the roof. And what this does is it's poisoning the nanopore system for doing fundamental biology. And I think that's just going to blossom now, because accuracies that are highly competitive with native RNA, there's no messing around with cDNA, yada yada. You're taking the RNA directly out of the cell intact, is going to be a sea change. And so I see in the next 10 years, particularly in molecules like tRNA, which is so heavily modified, it's the most abundant RNA species in the known universe, is something that you can do with Nanopore, and it's going to be extraordinary in the next 5 to 10 years.
So that's your wish list for Clive's talk tonight, is something nice on RNA.
Tyson RNA, and another one.
Yeah, no, that's similar for me. Having more accurate RNA sequencing from direct RNA, something that comes up a lot when I'm talking to virologists. They really want to be able to sequence the subgenomic RNA as well as the genome and see what different versions of the subgenomic RNA exist in the sample, with having that accuracy as well and having the throughput there. So improvements to that will please quite a few people in my field.
Well, keep your eyes peeled for Clive tonight. And just before we go, John, what would you like to see in Clive's talk tonight?
I guess I'm more on the side of, a new kind of simplification of library prep type, like diagnostic type.
Everyone
throughput.
agrees with that. Just so pass it on to Clive. Everybody in the panel says
simple library preps and low throughput diagnostic stuff.
Well, that gets us close to anything, anyone, anywhere. And Rosemary, in her opening talk tomorrow, is going to be talking a lot about end-to-end workflows as well. So we're excited to share that.
Okay.
Well, I'm afraid we've run out of time, but thank you so much for joining us.
It's been great.
And for the online audience, you've got a real treat of an afternoon coming up. We've got Cecil, who's going to be sharing the applications team. We've got the Nanopore Origins panel. We've got a wonderful early research scientist sharing their work from the beginning of those career journeys. So stay online and have a great afternoon.